Welcome back to Chapter 14, Part 3, and let's move on with translation. Translation is the second of the two steps in the making of a polypeptide. We talked about transcription before, and remember in bacteria, transcription can happen at the same time as translation, but in eukaryotes, it has to happen in the cytoplasm, so that has to have its own very separate step. And we have to, of course, modify our RNA in eukaryotes before we can get to translation. Translation itself is a pretty complicated process with multiple steps and multiple players. So let's start to get to know some of the players in the process of translation. A cell will translate mRNA into a protein with the help of another kind of RNA molecule called a transfer RNA, also known as tRNA. tRNA transfers amino acids to the growing polypeptide chain in the presence of a ribosome. So if we look over here, we've got all the players that we're going to get to know in a little while. We've got our messenger RNA here, right? It's the one going through the center. And we have our ribosome. There's actually two parts to the ribosome, the top part and the bottom part, or we say the small subunit and the large subunit. And then, of course, we have our transfer RNAs. And those transfer RNAs have amino acids on them, like right here. After it drops off its amino acid, then it doesn't have an amino acid anymore. And it'll go back into the cytoplasm. It's already in the cytoplasm, but it'll go back and look for its own amino acid, the same kind that it was carrying before. Again, translation is a complex process, and there's lots of biochemistry and mechanics, and so let's start to get to know that a little bit. First, let's talk about the structure of the transfer RNA. So transfer RNA is made up of RNA. That's the basics. And your book does give a lot of detail about this. So I want you to notice in this first diagram that we have the letters A, U, C, and G of RNA. So we know it's RNA. It has this very specific shape because it's held in place with these loops and these bends with hydrogen bonds. And of course, there are these three main regions, or I guess there's four. You really need to think about four main regions. The two most important regions are the one at the opposite sides, where one has a place where the amino acid can attach, and the other side has something called an anticodon. The anticodon is very important. We'll talk about this a little bit more later, but the anticodon is important because it does read the codons. And the amino acid attachment site is important because that's where the amino acid attaches. You can see transfer RNA sometimes depicted in this way, where it shows all the backbone and the bases. But the most common way is this little cartoon here, where it shows just the anticodon and then a little bit of folding, and you can see the amino acid attachment site. So this is the one that we're going to be really looking at as we're going through and learning about translation. So tRNA is just a single strand of RNA. Again, going back here, you can see it's a single strand. It's just folded back on itself a little bit, but it's just one strand with a 5' end and a 3' end. And it is about 80 nucleotides long. It can pair with itself with hydrogen bonding. And if you flatten into that plane, it does look like a clover leaf, but usually it's very three-dimensional, and so it looks more like this kind of L-shaped thing. And on one end of that L, it's got the amino acid attachment site, and on the other end of it, it has our anticodon. The anticodon is so important because it's the anticodon that base pairs with the codon in the messenger RNA. So in order for translation to happen accurately, the first thing that has to happen is that a transfer RNA has got to be matched with the proper amino acid. And this is going to be done by an enzyme, or actually a series of enzymes, called aminoacyl tRNA synthetase. So aminoacyl, it's going to add an amino acid to the tRNA, and that's what it does. It's going to synthesize this thing. So aminoacyl tRNA synthetase. And here's an example of aminoacyl tRNA synthetase, specifically for tyrosol. It's what attaches tyrosine to the tRNA. And it'll only attach tyrosine to a very specific tRNA base. So, this correct match is very, very important. Again, we only have 20 of them, so 20 amino acids, but we have 64 transfer RNAs, so we have to make sure that each time our tRNA is being added, the correct amino acid to it. Of course, it's also very important for there to be a correct match between the tRNA anticodon and the mRNA codon. Now, we do have this idea of flexible pairing at the third base. I never said this to you earlier, but it is kind of interesting. I pointed it out, but I didn't use this term. And the term I'm going to use is wobble. I'm going to go back up to this slide that I mentioned it, and we'll see what wobble really is. So if you look here, I was talking about CCU, right? CCU codes for proline, but so does CCC and CCA and CCG. All of those code for the same amino acid. And coding for the same amino acid, because the third base differs, that's what wobble means. It allows some tRNAs to bind to more than one codon. And so, yeah, CCU could attach, but maybe CCC could attach. You'd still get the same exact amino acid. Ribosomes are the place where the action occurs, right? It's where the tRNA brings the amino acid and it matches the anticodon to the codon in the mRNA. And so all of this is protein synthesis. Now, there are two parts to the ribosome, as I mentioned earlier. There's the large subunit and there's the small subunit. And both of the subunits are made up of both protein and ribosomal RNA. So you got some protein and you got some rRNA, ribosomal RNA. In bacteria and eukaryotes, ribosomes, the large and small subunits join to form the thing that is known as the entire thing. It is the ribosome. This whole thing is the ribosome. And these two things only attach together when there is also a messenger RNA kind of in the middle. And here you see, I'll just trace it with my pointer here. There's the mRNA. You can see that it goes through here, and it does attach kind of really close to these transfer RNAs, which we'll talk about in a second. Here's a more cartoonish version of the large subunit and the small subunit kind of attached to each other to make this very big ribosome. Notice that there are three locations in the ribosome, the E, P, and A site. I'm actually going to talk about this in a little bit different order. We're going to start out with the P site. That's the middle site, okay? And the middle site holds the transfer RNA that carries the growing polypeptide chain. So that's the P site. It's the middle one. The A site is where a new transfer RNA is going to come in. It's reading the codon, and it's going to transfer it's actually going to move, kind of slide over here to the P site. And when it does that, it's going to accept onto the end the growing chain. So here's what's going to happen is the transfer RNA in the P site is going to pass this along to the transfer RNA in the R site as it slides along. The E site, that's kind of like the exit site. So the exit site is where the tRNA that has lost its amino acid because it's now in the polypeptide, that's where it exits the ribosome. So we've got our 3 sites, each with a different job, each doing very important job, and of course the important job here is that we are making a polypeptide. So there are 3 stages to building a polypeptide. This sounds an awful lot like the 3 stages in transcription. It was initiation, elongation, and termination. So the stages are called the same, but of course they're doing other things. All 3 stages do require protein factors to aid in the translation process, and we are getting energy to make these chemical bonds. But instead of using ATP to do this energy, we're actually using GTP, guanine triphosphate, as the energy source of the building of the protein. So the initiation stage of translation happens when we basically build this ribosome and the RNAs all together. But it does happen in separate stages, so even the initiation stage is complicated in translation. So the initiation stage gets 3 sub-stages. The first part is where the small ribosomal subunit binds onto the messenger RNA, and the initiator tRNA, and there you can see that term right there, the initiator tRNA, scans along this RNA until it finds the start codon. Now the start codon is always AUG, and the amino acid that's found on the start initiator tRNA is always MET or methionine. So again, what we have in this first step is the mRNA binds to the small subunit of the ribosome, and then our initiator tRNA comes along and scans along till it finds the start codon, which is AUG, and that's the first step. The second step is where the large subunit, the large ribosomal subunit, comes along, and it kind of hooks up to the small subunit so that the initiator tRNA is found in the P site. Okay, so that's the second step, is the arrival of the large. One of the things that's really important is to understand is the start codon. It's extremely important. It's found in all RNA, all messenger RNA, because it establishes the reading frame for the growing protein. So they always start with AUG, and then each codon is 3-letter groups after that AUG. The addition of that large ribosomal subunit is the last thing that happens, and then we have these things called initiation factors. And initiation factors are just described, and they're not even pictured here, but it brings everything together. Notice that GTP is our energy source that happens and is going to be used throughout the protein building process. All right, so we're moving on to the elongation phase. We're actually making the polypeptide chain here. So during the elongation phase, amino acids are being added one by one by one to the previous amino acid. This happens at the C-terminus of the growing chain. And I just threw this picture in here to remind you that amino acids have 2 ends. They have an amino terminus and a C-terminus, an N-terminal and a C-terminal. And that's true for every single amino acid in this growing chain, right? So when you add a new amino acid to the growing chain, you're going to add it over on the C-terminus. Each addition involves proteins called elongation factors. So elongation factors is our concept here. And this does occur in 3 steps: codon recognition, peptide bond formation, and translocation. And I'm going to show those to you on the next couple slides. So again, codon recognition, peptide bond formation, and translocation. Translation does proceed in the 5' to 3' direction. So here we go. Elongation of the process of translation. I said the first part was codon recognition, right? So the codon comes into the A site, and the anticodon matches up to the codon. Again, you could have a little wobble there, but at the third base in the anticodon, that's okay, because it'll end up coding for the same amino acid anyway. So it comes in, and the transfer RNA binds to the codon. And the second step is called peptide bond formation, and here the long chain of amino acids is going to be transferred to the transfer RNA that's found in the A site. When that happens, we have something called translocation. And by the way, that whole process needed a little GTP energy. So for translocation, whatever was in the A site, now it has the chain, and whatever was in the P site, it's naked, doesn't have an amino acid. Everything slides along one step in the ribosome, and so the one that was in the P site ends up in the A site. And of course, once it's in the E site, it leaves, it exits, and then that leaves space for another transfer RNA to recognize the codon. And so we just keep going around this circle, using up 2 GTPs for every time you go around and add 1 amino acid. The third step in translation is termination. And termination happens when a stop codon is reached. Stop codons are found in the mRNA, just like all codons are found in mRNA. And when a stop codon reaches the A site of the ribosome, the A site accepts a protein called a release factor. Now, the release factor is an awful lot like a transfer RNA, but instead of having an amino acid at the tip, it has some water. It has a water molecule at the tip, at the place where the amino acid should be. It holds a molecule of water. And this reaction where the molecule of water is joined to the carboxyl terminus of the C-terminus of the amino acid, that's the end. There's no amino acid can join on after that. And so basically, termination involves just letting everything go. As long as this release factor comes and matches in the stop codon. And remember, there are 3 stop codons. Then this releases this polypeptide. We call it now a free polypeptide because it's not bound to a transfer RNA anymore, or the release factor for that matter. And everything releases. The large subunit releases from the small subunit, and the messenger RNA is released, as well as this release factor that no longer has a water molecule on it. This does require a couple of GTPs for the energy of releasing that material. But we're not really done yet, because although translation has been terminated, we still have got to make a functioning protein. And we're going to do that by doing a couple of different things. So translation in and of itself doesn't tend to make a functional protein. There are 3 basic things that can happen to a polypeptide chain after it's released from translation. It can be cut into little bits. And all of this, by the way, is called post-translational processing. So it can be cut up into bits, it can be little bits of sugar can be added. We talked about blood types and how that has sugars added to that, or it could have phosphates added to that. So there are two basic types of ribosomes, remember. There are ribosomes that are free ribosomes, and free ribosomes are found in the cytosol or cytoplasm. They're just kind of floating there in the cytoplasm. There are also bound ribosomes, and in eukaryotes, the bound ribosomes are found attached to the endoplasmic reticulum. Free ribosomes have an important job. Their job is to make proteins that are going to be functioning in the cytosol or cytoplasm. For example, maybe they're making the cytoskeleton, or maybe they're proteins involved in construction of ribosomes themselves, or making peroxisomes, or maybe some of the proteins that are used in the nucleus. So if a protein is going to stay inside the cell, inside the cytoplasm, it's going to be made only by the free ribosomes. If, on the other hand, the ribosomes are bound to the endomembrane system, they are going to make proteins that are maybe secreted from the cell, or maybe be put into lysosomes. Those also can be made by the endomembrane system. But if a protein is bound to leave the cell, if it's about to become part of the plasma membrane, or maybe it's a kind of hormone that's going out into the bloodstream, all of those are going to be made by the endoplasmic reticulum bound ribosomes. So the bound ribosomes. Polypeptide synthesis always starts in the cytosol, and this process is going to continue in the cytosol unless that ribosome is signaled by the messenger RNA, unless it's signaled to go and head towards the endoplasmic reticulum. If that signal happens, then it will finish up by putting that protein into the inside of the endoplasmic reticulum. So again, it always starts in the cytosol. It always starts at a ribosome floating around there in the cytoplasm. The messenger RNA binds to the small subunit and the large subunit, and we've got our initiator tRNA and all this other stuff. But if the tip of that polypeptide, if that has the right information, it's going to signal to a signal recognition particle. And so this signal peptide at the end of the long strand of polypeptide protein, if it signals this signal recognition protein, then that SRP then pulls that ribosome to the endoplasmic reticulum, and that protein is then strung and continue to be made inside of there. Now, interestingly, that signal peptide is going to be removed from the protein, and it's going to be removed by one of the pores that are located on the membrane of the endoplasmic reticulum itself. So again, that SRP, that signal recognition protein, allows that ribosome to be bound to the endoplasmic reticulum. And it's bound to, I said it was bound to a protein, but that protein is, in fact, it's a receptor. Polypeptide synthesis is continuing, and it's going to happen to the inside, and the inside of the endoplasmic reticulum is called the lumen. So I hope that makes sense. We've got to have that additional step happening if we're going to make proteins that are headed out towards the plasma membrane. like any of those recognition proteins that we talked about before on this plasma membrane. And again, often protein-based hormones are going to also head out. As we talked about before, when we were talking about bacteria can make lots of proteins at the same time that it's making the RNA. So this can happen in both bacteria and in eukaryotes. And it happens by this process called a polyribosome. We also call it a polysome. And actually, this is the term that I tend to use, is the polysome word. But polyribosome also works. Polyribosome actually makes a little bit more sense because we're saying many ribosomes are on an RNA, LL at the same time. So if this is where this is the 5' end of our mRNA, the ribosomal subunits come in and they bind to that. Don't forget about our initiator tRNA, and then it starts reading the mRNA and making proteins. And this is a very, very fast way to make a whole bunch of the same protein that you might need. This is an actual electron micrograph showing the polysome, or the polyribosome, depending on how you say it, along a single mRNA strand. Also, again, remember that in bacteria, transcription and translation can happen at the same time. So clearly, things can get pretty interesting. In this picture, this strand right here is the DNA. Okay, and each of these little strands coming off, that's RNA. And then on those RNA are polysomes, right? Are polyribosomes. So DNA is being made by lots of RNA, and here, right here at the tip of this one, you can see the RNA polymerase. And then as the RNA polymerase reads, here's another one in this light gray here. As the RNA polymerase reads down the course of the DNA, it's spitting out the RNA, and then the ribosomes come on and bind onto those, and you get polyribosomes. And you spin out here, we can see very nicely, proteins. Now, the protein, and this is a little bit counterintuitive, because as you go along the DNA, your RNA gets longer and longer. That makes sense. The thing that's a little less counterintuitive is the polysome. So let's take a look at the longest RNA and notice that the one down here has the shortest amount of protein coming out of it. And that's because it just grabbed on. The ones up here have been reading all the way up through here. So it's going to have a much longer protein than the ones that are closer to the 5' end of the RNA. So putting it all together, making multiple polypeptides in bacteria and in eukaryotes can involve transcription and translation of the same mRNA. So multiple RNA polymerases can read the DNA at the same time, making multiple mRNAs from the same gene. In bacteria, you can do translation at the same time. In eukaryotes, you got to leave the nuclear envelope. Also, remember, you had to go through some RNA processing. You got to add that cap and tail and splice out the introns and join up the exons. But all of the translation is going to happen in a eukaryote. It's going to happen outside of that, outside of the nucleus, in the cytoplasm. Well, that's all that I have for you for part 3. We'll continue with part 4 in just a moment.